Hello.
Hello, yes, sir. Good morning.
Good morning.
Sir.
Am I audible?
Yes.
Am I audible?
Yes, sir.
you
Yes.
are audible.
Okay.
Hi, this is Larissa here.
Hello, ma'am. Good
ma'am.
morning.
Good morning, ma'am. Hello.
Anupama, ma'am, we can start with session.
Okay. Good morning, one and all present here. It's a lovely Monday morning, and as rightly said, you don't have to be great to start, but you have to start to be great. I, Mrs. Anupama Tomar, welcome you all for the international webinar Biofacet, organized by the Department of Microbiology and Biotechnology at Patkar-Varde College. I extend a warm welcome to the guest speaker, Ms. Larissa Falcao, our CEO, Dr. Mala Kharkar, our principal, Dr. Shrikant Sawant, Chief Coordinator, Ms. Ruchita Rane, department staff members, and all our dear participants. Department of Microbiology and Biotechnology has been organizing Biofacet every year. Started in the academic year 2012-13, Biofacet is a stride taken by the department to provide a platform to scholars, teachers, and experts from various fields of science and technology for academic interactions, elevation, and enrichment. It has also taken initiatives in helping students in extending their minds to diverse fields available in biological sciences, and since last year, Biofacet has taken the shape of national conference. This year, we are extremely glad to take Biofacet to an international level. This would have not been possible without your support and participation. Thank you all for all your support. We are glad to have an overwhelming response of more than 300 participants gracing us today. The topic of today's webinar is vaccine development process. And as we all know, that vaccination is a part of multifaceted public health response for the future emergence of a pandemic illness. In addition to other measures which are designed to respond and control a pandemic, such as surveillance, communication plans, quarantine, and disease treatment, deployment of effective vaccines has the potential to protect lives and limit disease spread. Not all disease threats, however, have a corresponding vaccine, and for those that do, there are significant challenges to their successful use in the pandemic. In this unprecedented times, It is required that all countries, cities, and communities join the fight against COVID-19 and develop successful vaccine which will serve as a relevant weapon in this pandemic. As Nelson Mandela said, life and death for a young child too often depends on whether he or she is born in a country where vaccines are available or not. We are extremely fortunate to have a renowned researcher, Miss Larissa Falcao, ma'am, with us today to share her experiences on vaccine development. Miss Larissa Falcao has completed her bachelor's and master's in pharmaceutical sciences from Mumbai Educational Trust Institute of Pharmacy and Drexel University College of Medicine respectively. Highlighting her endless list of achievements, she has worked with Dr. Paul Campbell Laboratory and Dr. Alessandro Fatatis Laboratory on signaling of the RAF effector pathway downstream of KRAS in human pancreatic carcinoma. She has worked with Hemisworks Biopharma as research biochemist and with Pfizer as senior research scientist. Currently, she is working with Pfizer, New York, and her research areas involve clinical trials on vaccines. She has monitored human and cellular immune responses to respiratory syncytial virus. The vaccine is currently in phase 3 clinical trial. Also generated synthetic DNA constructs for influenza vaccine. Again, currently it is in preclinical trial and presently working on de novo gene synthesis platform to generate DNA constructs for the COVID-19 vaccine. Currently in preclinical trials. She has also received several W.E. Upjohn Above and Beyond awards at Pfizer for her contributions to the COVID, influenza, as well as RSV vaccine. Currently on track to receive a team award for outstanding and prompt generation of vaccine constructs for the COVID-19 program. She has also received the honor to give a short talk on the COVID-19 special episode on Vivid Bharati. Now, I would request our principal sir, Dr. Shrikant B. Sawant, to address the webinar and declare this international webinar open.
Thank you. Very good morning to all. This gives me immense pleasure to welcome all the participants in this international RUSA-sponsored international webinar organized by Department of Microbiology and Biotechnology of Chikisak Samoa's Patkar-Varde College. I also welcome all the dignitaries and our eminent speaker, renowned scientist Larissa Falcao, who is working with Pfizer. Our college was founded by Chikisak Samoa, an association of like-minded individuals who were keenly interested in education. The college was named after Sir Sitaram Patkar, a distinguished judge of the Bombay High Court, who was knighted after his retirement, and his wife, Lady Shantabai Patkar. Sir Sitaram was deeply interested in furthering the cause of women's education and was the Vice Chancellor of the SNT University for many years. The College of Arts and Science started functioning from the academic year 1964-65 and the Commerce section from the academic year 1980-81. To provide career-based education, the college started degree-level self-finance courses from the year 2000-2001. Since then, the college has constantly upgraded the courses offered and the infrastructure required for these courses to provide students with the best possible learning opportunity. In keeping up the spirit of learning and growing through criticism, we have set up high standards, working continuously towards advanced goals. Many of our alumni hold very good position in various fields. Patkar-Varde College has a place of pride among us the various educational institutions in the western suburbs of Mumbai. Our college is re-accredited by NAC in its third cycle with A+ grade, securing 3.53 CGPA. Autonomy was conferred on the college from the academic year 2019-20. The college is recipient of Best College Award by the University of Mumbai for the year 2016-17. As per Education World India, college ranked 45th in all India and 10th in Maharashtra among the private autonomous colleges. Today, on this occasion, it gives me immense pleasure to welcome the renowned scientist, Miss Larissa Falcao, who will share her experience on vaccine development process. Friends, we know that in this pandemic situation, everyone is looking at vaccine as a boon to control this pandemic situation, to control death, to control havoc. Vaccine development. There are a number of companies trying over. And what is vaccine development process? What stages are there? How research is involved? How clinical tests are conducted? On all these issues, I'm quite sure that today's eminent speaker, scientist, will throw light on these issues. On this occasion, I also welcome our Honorable President, our Honorable President, Sri Kishorji Varanganekar, sir, our Vice President, Rajesh Desai, sir, Dr. Gurunath Pandit, our Joint Secretary, Dr. Sri Girish Amonkar, Joint Treasurer, and all the office bearers of Chikitsak Samuha. I also welcome Mala Kharkar, Madam, our CEO, and on this occasion, I declare this international webinar is inaugurated. I wish you all the best. I'm quite sure that the participant will be delighted and enriched with the knowledge on vaccine development process. Thank you very much.
Thank you, sir. Before we begin with the webinar, I request all the participants to remain patient. If any technical problems arise during the webinar, we will try to resolve all the technical glitches. Kindly post your queries in the chat box. It will be answered at the end of the session. It's a humble request to kindly maintain the decorum of the webinar. We are sure that this webinar would certainly complement to your learning objectives. Now, I would request Larissa ma'am to address the webinar.
Hi, good morning, everyone. I hope you can hear me clear.
Yes, ma'am. Yes,
Okay.
ma'am.
Yeah. So thank you, Dr. Sawant and Mrs. Anupama Tomar for the beautiful introduction. It is my great honor to be presenting today in front of such a diverse audience. Today I will be talking about the process of vaccine development in brief. Before I begin, I just want to say that since I work for the pharmaceutical company Pfizer, and since Pfizer has been involved in the recent COVID vaccine development effort, any statements that I will make today will not be on behalf of my company, but they will all be personal statements. I also want to point out that all of my slides are numbered on the right bottom here. So if you would like me to go back to a particular slide during the question answer session, you may note the slide number. So with that, I would like to begin. So what is a vaccine? a vaccine is a preparation that provides active immunity to a particular infectious disease. And it typically contains an agent that resembles the disease-causing microorganism itself. To give you some historical perspective of where this process of vaccination began, as early as 10th century, inoculation was a pretty established procedure. Some scientists from China or Egypt had carried out this procedure of inoculation, where they would take infection from an infected person and give it to a healthy person such that that person would achieve immunity. But in the 1700s, Edward Jenner, a renowned physician and scientist, he's the one who actually developed a protocol and established the actual process of vaccination. So now in 1700s, there was a great epidemic of cowpox and smallpox happening at the same time. And it was noted that some of the milkmaids who were attending to some of these cows that were infected with cowpox started getting the cowpox virus and getting lesions on their skin. It was also noted that subsequently they would become healthy again from these lesions, but then these milkmaids would not be subsequently infected with smallpox. They were somehow immune to the smallpox virus. So a lot of scientists made these observations, and it was Edward Jenner who took the infection from the wounds of Sarah Nelmis, who was such a milkmaid who attended to some cows which had the cowpox. And he gave this infection to an 8-year-old boy, James Phipps. Now, James Phipps became infected with this cowpox from Sarah Nelmis. And after falling sick with a mild case of cowpox, he eventually recovered. Then Jenner went to the next step, and he took scabs from a smallpox patient this time, and he infected Phipps again. But this time, as he expected, Phipps was protected because of a prior exposure to cowpox, and therefore his protection was complete. This was a first established vaccination experiment. And this is why the Latin word for cow is vaccine. And that's why we now call all this process of inoculation as vaccination. I want to take you through the basic principle of vaccination. So in vaccination, an antigen, which is usually derived from the pathogen itself, by one form or another, and I will be talking about that in the subsequent slides. This antigen is injected into the body of the human. In the body of the human, then antibodies will be generated contrary to this antigen. These antibodies will be circulating in the body of the individual. On exposure to the pathogen, these antibodies will bind to the pathogen and cause destruction of the pathogen or its neutralization. Now, this is a short-term lived response in most of the cases, but whenever a person is vaccinated or whether he is exposed to a pathogen, there is also a memory response that is produced in that person's body that will be more sustainable, may go on for years or for months. And in case of a further exposure of a pathogen, this memory response can also be triggered to go back and cause the destruction of the pathogen. Now I'll take you through the types of vaccines. So these are the 4 different major types of vaccines that are currently being made. The first 2 categories actually involve the entire pathogen itself, the entire bacteria or the entire virus. In the first case, they take the live virus and just weaken it with chemical treatment or heat treatment. An example of this is the oral polio vaccine. I'm sure most of us have received this oral polio vaccine doses when they were young. So that's an example of a weakened form of the polio virus strain. A similar approach, but this time the entire virus or bacteria is killed, is also used. And that's the approach that's used in the inactivated polio vaccine, which is an injectable formulation. A third kind of vaccine is an inactivated toxin, or it's also called toxoid vaccine. In this case, it protects us from the harmful effects from the infection and not due to the pathogen itself. To give you an example, in case of tetanus, you must have heard of tetanus. So this is a disease that is caused by the infection of Clostridium tetani, which is a bacteria. When Clostridium tetani infects a person, it releases certain toxins, which leads to tetany, which is muscle pain and joint pain. So if you give a tetanus toxoid vaccine, it will produce antitoxins against this toxin that the Clostridium releases, and therefore it will neutralize and help you to fight the tetanus. The final type of vaccine, and which is more broadly used recently, are biosynthetic vaccines. And these involve proteins or peptides from the surface or other parts of the pathogen itself, or its genetic material, which is the DNA or RNA. An example of this would be the hepatitis B vaccine, which also I'm sure many of you must have received the 3 shots of the hepatitis vaccine. This is a recombinant protein vaccine. Now I want to take you through the transmission of an infectious disease. So the infectious disease transmission is not as easy as if one person gets infected, he gives it to another, who gives it to another, who gives it to another. It's more complicated than that. The transmission of an infectious disease follows an exponential pattern, which means one person can give it to 2 more people, who in turn can give it to 2 more, who in turn can give it to 2 more, and the cycle goes on. Now, if recently, if anyone has invited you to a party or a wedding or a social, and if you didn't go, this is the kind of impact that you may have generated. So thank you very much for social distancing. I also want to explain the concept of herd immunity a little bit. So on the left side here, assuming all of the members of a particular population are equally susceptible, if there is an individual that is infected in that population, he is capable of infecting many others. But in case of a population where a majority of the people are vaccinated, the spread of such infection is very short-lived. And therefore, we need this kind of a situation of herd immunity, which can be achieved by a vaccine, so that we do not have a pandemic-like situation. Now, these are the different approaches to a COVID-19 vaccine that have been used in the recent past since the beginning of this pandemic. To give you a brief, this is how the coronavirus looks. So it's a circular virus with RNA genome, and it has these proteins on the surface like a crown, which are spike proteins. And as you can see in this RNA, there is this red region. That's the code that codes for this spike protein. Now, like I mentioned in my previous slide, these several approaches are available for types of vaccines. So similarly, for the COVID-19, there are some live attenuated or inactivated vaccines available, where they take the entire pathogen and just make some minor changes such that its infectivity is reduced, and they use that as such as a vaccine. There are also some genetic approaches that are being used. For example, they take an engineered DNA molecule and they add the code for the spike protein into that engineered molecule, and that is given to the host. And when this DNA enters the host, that DNA will be converted to mRNA, and that mRNA will be converted to the spike protein. And then that spike protein will act as an antigen, and antibodies will be generated against that. Another similar approach is that of an RNA-based vaccine, where they use like a lipid molecule or some other kind of basis, and they introduce an RNA itself into the host. This is the kind of vaccine that is currently being developed by Pfizer and Moderna and some other leading pharmaceutical companies who are at the top of the COVID-19 effort. A third kind of genetic approach is taking a viral vector, which is not again infectious, and several kind of viruses are available that could be used for this approach. For example, adenovirus. So you take the vector and kind of use its ability to go inside the cell, and you put the genetic material that you want to be translated into protein inside this vector, such that this vector will act as a host and get your mRNA entry into the cell again. And then the final approach that has been used, this is also known as a virus-like particle. So if you can see, this is these are spike proteins stuck again onto a lipid surface, but it resembles very closely what the coronavirus looks like. So they are called VLPs, or virus-like particles. And this final approach that also has been under development by a few companies is recombinant proteins. So they are just portions of recombinant spike proteins that will be either given with an adjuvant or a vehicle, and thus provoke an anti-spike protein response. So since I am more familiar with RNA vaccine technology because I have been working on that, I would just like to take you briefly through how RNA vaccine technology works. So like I mentioned before, the RNA coding for a specific antigen is used. In this case, the spike protein code is used, and it's injected into a human. Once injected, it enters into the cell, and then this RNA is converted to the protein, which will act as an antigen, in this case, the spike protein. So this spike protein will get presented on the surface of your host cells, and then the immune cells will recognize that as a threat and develop these antibodies against it. This is how an immune response will be generated in response to an RNA vaccine. Of course, there's also going to be the memory factor, which I explained to you before. Such kind of an RNA vaccine technology holds a great promise for improved personalized medicine for cancer in the future. So for example, if there is a particular tumor, to do tumor analysis, you can identify what specific antigens are present in that particular individual. And then from those antigens, you can find out the genetic code and manufacture RNA coding for these specific tumor antigens. These will then be injected into the human such that all of these specific RNAs will code for these specific antigens, and therefore it will be a more direct immune response. So there will be not any non-specific response that we have seen in, for example, in chemotherapy, because chemotherapy attacks rapidly dividing cells. But since here it is the specific target, so only the tumor cells will be killed. So that is a great promise for the future. So now I want to take you through the concept of adjuvants. An adjuvant is something that is used with vaccines to help create a stronger immune response. So I showed you these different approaches in my previous slide. This is the coronavirus, and then this is the live attenuated, some genetic vaccines, and some protein based. If you go from left to right here, there is an increased tolerability in these vaccines as compared to these here, but there is a reduced immunogenicity, and that's just because this large chunk looks like a stronger danger signal to the body, whereas these more purified protein bits do not look like such a great danger signal. So what adjuvants do is, adjuvants are tagged along with these proteins such that it makes it look more bulky, and it makes it look like a larger danger signal to the body, such that a good amount of immune response will be mounted against it. Now, the adjuvants don't necessarily have any effect of themselves, and that is tested in any of the prior trials before using these adjuvants. So these are just some examples of different adjuvants that are currently being used. Aluminum is one which has been used in many vaccines. I'm sure if you must have heard a few of these vaccines listed here, like Trumemba for meningitis B or Prevnar 13 for pneumococcal pneumonia, etc. So aluminum is known to again have this adjuvant effect and increase the immunogenicity of a particular vaccine candidate. There are some other adjuvants which are also present. For example, the ASO1B and CpG. These are kind of lipids or fat-like molecules. So in addition to having the adjuvant effect, they also act as the vehicle itself that carries this protein or RNA molecule inside of the cell to generate the immune response. So it's kind of a dual mechanism of increased immunogenicity as well as acting as a vehicle. This is just a slide to show you what effect the adjuvant technology has had on vaccine development. So since the first vaccine was made by Edward Jenner against smallpox in the 1800s, if you can track by time, you can see that the blue dots, which is the adjuvanted vaccines, there are more of the adjuvanted vaccines recently since the 1980s. So this is the effect of adjuvants in the vaccine technology. More and more vaccines are getting approvals. So now I would like to take you through the general stages of development of a vaccine. I would just like to say that since I've been working on vaccine development in the United States, these stages are more relevant, and the approval agencies, etc., that I will be talking about, the FDA and the CDC, they're more relevant to the United States. But in every country, similar stages have been done for a vaccine development. So first, I would like to take you through the exploratory stage. This stage usually takes approximately 2 to 4 years. It is typically carried out by federally funded academic and governmental scientists. The government usually gives funding to the scientists to carry out this kind of an exploration. And it's basically to identify natural or synthetic antigens that will help prevent a disease. To give you a few examples of what happens in the exploratory phase, here is, if you think about the target pathogen influenza virus, then the exploration will involve what kind of proteins are there on the surface which are integral to the infectivity of the influenza virus, such that we can make a vaccine against it. So in case of influenza, it's the hemagglutinin or HA protein. And then, of course, you all must have known that for coronavirus, it's the spike protein. So just this kind of investigation is done in the exploratory stages. This is an this is a transmission electron microscope image of SARS-CoV-2. It's arising from the cells, surface of cells cultured in the lab. And these spikes that you see on the surface are actually the spike protein. And these spikes actually give coronavirus its name because it looks like a crown. So coronavirus. This is another colorized scanning electron micrograph of an apoptotic cell in blue. So these cells are undergoing apoptosis, and these red dots are the coronaviruses that had infected these cells, which are now emerging on its surface. This is some structural modeling that has been done to understand the interaction of the ACE2 receptor in the host with the spike protein on the surface of the coronavirus. So this ACE2 receptor is present on human lungs and some other tissues and organs, whereas this spike protein, like I mentioned to you, is present on the coronavirus surface. This red portion of this spike protein is called receptor binding domain, or RBD, which acts as the main lever which binds to this ACE2 receptor. So through this model is how the coronavirus will enter into the host cell. So that's kind of what happens in the exploratory stages. The antigens are found, some kind of modelings are done, what kind of receptors are involved, etc. Then we move on to the preclinical stage. The time frame for preclinical stage is 1 to 2 years. It involves tissue culture or cell culture in petri dishes or in flasks or in plates. It also involves some animal testing. The purpose of this is to assess the safety of the candidate vaccine and also its immunogenicity, how immunogenic or how much of an immune response can the vaccine produce in animals. These studies give researchers an idea of the cellular response that they can expect in the future in humans, just based on the animal readouts. They may also suggest a safe starting dose for the next phase. So if you can do a dose escalation study in animals, then you will know what kind of a dose you could give in humans. And there are also some animal challenge studies, which I will brief you in a few slides. And of course, if at this point of preclinical study, a vaccine does not show the safety and efficacy that is expected of it, then it will be declared as a failure and it won't go to the next phase. So a lot of animals are used in preclinical research. The choice of the animal model depends on different factors. For example, the scale of the experiments, the costs involved, the susceptibility of the animal to that particular target pathogen, virus or bacteria, the pathophysiology of the disease, whether that particular model is showing the symptoms that we need to see for that particular virus or bacteria, and also the availability of the model. In the United States, there are many committees that are available for ethical treatment of animals in research. So all the protocols that have to be done on these animals are first reviewed by these committees, and when they're approved, only then are these animal experiments performed. So here I just want to give you a typical example of how a preclinical study looks like. So I just picked this paper and you can refer to it if you want to read in detail. But this is a preclinical evaluation of the immunogenicity and safety of an inactivated enterovirus 71 vaccine candidate. So human enterovirus is a virus that causes hand, foot, and mouth disease in young children in the Asia Pacific region. And what the authors of this paper have done is they've taken this pathogen itself and inactivated and purified it, kind of the first approach of the type of vaccines that I told you about. So they inactivated and purified it, and then they used an adjuvant, alhydrogel, which is similar to aluminum, which I showed you in the previous slides. They took this EV71 vaccine along with the alhydrogel and injected into mice. And then they collected the antibodies that were generated in the mouse blood at different time points. Then they took this antibodies and exposed them to live enterovirus and added this entire mixture onto cells. Now, if these antibodies are specific and if they are able to neutralize this particular pathogen, then these cells will not have any infection. But if these are not specific, then this pathogen will be able to infect this, and therefore it won't be neutralized. This typical experiment is very common in majority of the virology vaccine developments, and this experiment is called a virus neutralization assay. So these are the results that they saw for their virus neutralization experiment from their study that they did in mice. I would like to take you through the different groups that they use. So first, they use a PBS control group, which is just a saline control group acting as a negative control. They also use an alhydrogel control group, which means they only got the adjuvants and none of the antigens. And then here on the left, they use the EV71 at 3 different doses, 0.1, 0.6, and 3. And then the same 3 doses with alhydrogel. So these are the different groups that they use in this particular study. The vaccination was done at time point 0 and then time point 28 days, indicated by these red arrows here. And then this is on the x-axis is the days post the primary immunization. And on the y-axis is the neutralizing titer. So basically, the higher the neutralizing titer, the more efficacious a vaccine is. So if you can note, this EV71 going from the smallest dose to the highest, there is an increased amount of neutralization titer that is seen over time. And as much as after 91 days after immunization, they almost flatline and become a plateau. There is also this dose-dependent effect seen with the incorporation of alhydrogel, which is an adjuvant. So this is just a typical example of what is done in a preclinical study. People will look at the dosages that can be used, the 3 different that were used here, or will it be a 1-shot or a 2-shot kind of procedure? What kind of adjuvants will be used and are they necessary to be used? And also, of course, the efficacy. How much neutralization will these antibodies produce in that particular pathogen? Next, the same antigen was used in a toxicology study in rabbits. So they used the low dose of the 0.6 micrograms as well as a high dose, which is 3 micrograms, and they evaluated safety in this rabbit model. So no morbidity or mortality was seen in this model. There was no skin reactions that were seen at the site of injection. The hematology and clinical chemistry parameters also remained within the normal range, like the blood pressure, the temperature, etc. No difference in group averages of body mass or food consumption in these rabbits was seen. There was just one incident of muscular degeneration that was seen at the site of injection, but that was also with the alhydrogel placebo group and not with any antigenic group. And this also recovered within 2 to 4 weeks. So you see that this is the kind of safety evaluation that is done in a preclinical study. You take an animal model and you see what kind of systemic and local effects happen in that animal model. And again, if you want to refer this paper, there is more information at the bottom here. Next, I want to talk to you about challenge studies. So in this particular challenge study that I am talking about here, this paper, they took some mice and they injected PCV13 vaccine, which is a vaccine for Streptococcus pneumoniae. They injected the mouse at week 0, 2, and 4 with this vaccine. And then at week 5, they actually challenged this mouse with live Streptococcus pneumoniae bacteria. They use a strong concentration here, about 10 raised to 5. They also did the same kind of experiment, but this time here in the second case, they did not vaccinate the animal. They only challenged it with the Streptococcus pneumoniae. And this time the challenge was done, one with the same dose as the previous here, and also with 2 lower doses of Streptococcus pneumoniae. So on the bottom, you can see The x-axis is hours after this particular infection. And on the y-axis, you can see the survival rate of these particular mice. So this first line here with the solid dots is the one that the mice that did receive the vaccination. And you can see that 100%, there is a 100% survival rate over time for these mice. But if you can see all of the other groups, whether they receive the 10⁵ or 10⁵ or 10⁵ concentration of this Streptococcus pneumoniae, they all eventually in time succumbed and died due to that infection. So this is a typical example of a challenge study. So it is similar to what you saw in the neutralization assay in the previous study, but here the neutralization happens inside the mouse and not in a cell culture. So once the preclinical phase is completed, a sponsor usually, for example, a private company submits an application to the US FDA for an investigational new drug. The sponsor will describe what kind of manufacturing and testing processes will be used, will talk about the lab reports and describe the proposed studies that will be done downstream from here. The FDA will approve this application within 30 days. And then once the IND application has been approved, the vaccine is subject to 3 phases of testing in humans. So we went over the exploratory stage and the preclinical stage along with the IND submission. After the IND submission, we'll now look at the clinical stage of development. So this is the first stage of clinical development. Here, this involves a small group of adults, usually between 20 to 80 subjects. If the vaccine is intended for little children, then first adults will be tested and then they will gradually step down the age until the test subject age has reached their target age. Again, the purpose of this kind of a study is to assess the safety of the candidate vaccine, this time in humans versus animals in the previous. And also they want to look at the type and extent of immune response that the vaccine provokes in humans. The challenge model could be implemented in a minority of these clinical development phases, but it really depends on what kind of virus or bacteria you're using. If the virus or bacteria is too deadly, then a challenge model might not be feasible. And then again, phase 1 trials can be blinded or non-blinded. Blinded means that both the receiving person as well as the one who's administering the vaccine does not have information whether he is administering the vaccine or a placebo. So in this case, it can be either or. In clinical development, phase 2, the same kind of studies happen in this phase 2, but this time it involves several hundred subjects. And some of the individuals may belong to groups at risk of acquiring the disease. So for example, in the current COVID situation, if New York was the epicenter, then in this kind of a clinical development phase 2 study, they will involve more subjects from the New York area just because they have a higher risk of acquiring the disease, and therefore better judgment of the immunogenicity of the vaccine can be made in that case. These trials are randomized. So there are different sexes involved, different ethnicities involved, different backgrounds of individuals involved, and they are all randomized in different groups. They're well controlled. So there is a well established placebo group and a well established vaccine group. The purpose again is similar to the previous 2 studies to assess the safety, immunogenicity. There will also be some proposed dosage studying that will be done. And then the schedule of immunization and the method of delivery will be looked at in this phase. So I want to give you an example of what a phase 1/2 study looks like. So I picked this particular example because this is Pfizer and BioNTech's current COVID-19 candidate that is out there in phase 1/2 trials. What Pfizer did here is they screened 76 participants for this particular study, out of which 45 participants were enrolled and randomized. The others didn't meet the eligibility criteria. These 45 participants were further divided in 3 groups. The first group received 10 micrograms of their target RNA vaccine. The second received 30 micrograms of their vaccine, and the third received 100 micrograms of their vaccine. And each group was each group was included with a placebo. Now, in each of these groups, there was one dosage that was performed, and then some safety evaluations were done, and if they were found to be safe with the first dose, then an additional dose was used in all of these individuals. So this is just a study designed for the phase 1-2 of the COVID-19 vaccine by Pfizer. Some of the local reactions that were reported within 7 days of vaccination for all dose levels are reported here. So on the x-axis, there is pain, redness at the site of injection, as well as swelling. And then there are these different dosage groups. So for the 10 micrograms, the first shot and the second. For the 30 micrograms, the first shot and the second, and so on. So as you can see, if you look at this particular parameter, pain, the symptoms that are shown in all of these individuals are usually mild to moderate. So the subjects experience some kind of mild to moderate pain. Only in this 100 microgram group, on their first shot, about 10% of the population received some severe pain. And if you notice, that's why this particular group did not go through the next shot. So there is no dose 2 level available for this 100 microgram dose. And then for redness and swelling, there was not much reported for any of these groups. Next, they look at the systemic events and medicine use that was reported within 7 days of vaccination. So this is the dose 1 of each of these different strengths that was used, and this was the dose 2. And they looked at different parameters such as fever, fatigue, headache, chills, vomiting, diarrhea, muscle pain, joint pain, and medication use. And as you can see, there is pretty much a dose-dependent effect in the systemic events that has been seen. There is less of an effect with the 10 microgram and 30 microgram dose as compared to the 100 microgram dose. And then again with the dose 2, some similar observations were seen. So that was the safety perspective of that study. Now I'll take you through the immunogenicity perspective of the study. On this left panel here, this is again a neutralization assay or a neutralization experiment that was done. So basically these humans were injected with these different doses, the 10 micrograms, 30 micrograms, and 100 micrograms. And then at different time points, which are listed here on the x-axis, the antibodies, blood was collected from each of these humans, and then antibodies were purified from that blood. These antibodies were subject to coronavirus in a cell-based neutralization experiment. So basically on the x-axis, y-axis again is the neutralizing titer, and the higher the titer, the better the neutralization of the coronavirus that is seen. These arrows indicate the 2 shots that the particular subject received over time. So this first panel here is the 10 microgram dose. The second panel in darker green is the 30 microgram dose. And the third one is the 100 microgram dose. And as you can see, as I told you, based on the systemic events that were seen in the 100 microgram dose, there was only 1 shot given. The second one was discontinued. So as you can see, after the second shot, there is a pretty good amount of neutralizing antibodies that are generated in both the 10 microgram as well as the 30 microgram dose. And there is a positive control to this, which is the human convalescent serum. What they did here is they acquired serum from individuals that were infected with the actual coronavirus itself and saw how their antibodies do in neutralizing the coronavirus in the same experiment. So you see that this 10 microgram and 30 microgram dose after the 2 shots is able to produce a stronger neutralizing titer than even the human convalescent serum. So this data is very promising. They also did some binding studies here on the right, where they took these same antibodies over time and they looked at how well they bind to the spike protein. So this is the RBD binding domain of the spike protein. And so they just looked at how well these bind together just to get an estimation of binding. And such kind of studies are always done in the phase 1 and 2 trial, where you get an estimation of the binding versus the neutralization, because it's not always the case that if there is good binding that there will be good neutralization. You will have to look at both. So once that is done, you move on to the clinical development phase 3. So this time now it involves thousands to tens of thousands of subjects. And this is totally randomized and double blinded. So neither the investigator nor the subject who's receiving the vaccine knows whether he is receiving a vaccine or a placebo. And then at every step in this kind of a study, intermittent analysis is done. So for example, if there are 10,000 subjects, for every 1,000, the data will be collected and analysis will be done whether this particular study can go on or whether there are any warning signals or signs that are coming that we need to pause this kind of a study, because 10,000 subjects, putting 10,000 subjects at risk is a lot. The purpose of this kind of a study is to assess the safety of the vaccine in a large group of population. So for example, if there are certain rare side effects that are seen 1 every 1,000 individuals, but they are real side effects to this vaccine, then you will only be able to record such kind of side effects if you have a much larger subject population. So if 1 every 1,000, then you will at least need about 10,000 to get 10 events of this rare side effect. And that's the purpose of having such a large chunk before moving on to billions and billions of people which will actually receive the final vaccine. And of course, they will also look at the vaccine efficacy in this kind of a trial as well. So after the phase 3, the vaccine candidate moves on to approval. So a biological license application is submitted, a BLA, to the FDA. The FDA will look at the manufacturing premises, approve the labeling of the vaccine, look at all the procedures if they are in place or not. Once the license is approved by the FDA, there will be periodic inspection of the facilities where this vaccine will be manufactured. The manufacturers' test lots of potency, safety, and purity will be evaluated by the FDA. And then FDA also has the right to conduct its own testing on this manufacturer's vaccines. So the FDA can visit a particular manufacturing site, collect the samples, and do their own testing to understand if there is any cause for alarm. And then manufacturing capacity is a big thing in pharmaceutical industry as well. So for example, imagine you have to produce 50 billion doses of this coronavirus. To generate such a large amount, you will need a large manufacturing facility with great sizes of bioreactors and tanks. So that is also some of the considerations that need to be taken care of during this process. And finally, there is adverse event monitoring. So again, the government has this system called as Vaccine Adverse Event Reporting System, or VAERS. It's a national early warning system to detect the possible safety problems in US licensed vaccine. It is supported by and sponsored by the CDC, the FDA, and the Health and Human Services in United States. And it is a voluntary reporting system. So anyone like the patient itself, or a parent, or a healthcare provider, or a friend of the patient who thinks that there are some kind of adverse events associated with the vaccination, they can go to this VAERS system and report it. The CDC will then take this report and investigate the event and try to find out whether this adverse event was in fact caused by the vaccination or it was just a one-off. There is also something known as the Vaccine Safety Data Link, VSD. It is a collection of linked databases containing information from different medical groups. From different clinical studies, what kind of information is available will be put into this safety data link, safety-based information. And then whenever a pharmaceutical company wants to run a particular kind of trial, and if they're using a particular kind of adjuvant or a particular kind of vehicle, they can request to access this database and see what kind of data is out there for that particular adjuvant. And then finally, there is the clinical development phase 4. These are optional studies that the drug companies may conduct after the vaccine is released. It is basically to further continue to test the vaccine for safety, efficacy, any other potential uses, off-label uses, etc. So I took you through all of these phases, right from exploratory stage, where you kind of understand the antigen that should be going into the vaccine, the preclinical stage in animals, the clinical stages in humans, and then the manufacturing, regulatory review and approval, and the quality control. So on a lighter note, I just want to tell you some of these annoying questions that are posed to scientists recently. A few of them are, when will the COVID-19 vaccine be ready? What is taking so long? Why don't they just start vaccinating with what they have and we'll see what happens? So I just want to remind you what I took you through in my talk today. This is what happens in the exploratory phase where you understand the antigen more. This is some of the safety efficacy studies that need to be done in animals. Then you have to do some toxicology studies to understand if these vaccines are safe in animals. The same kind of studies are then done in a smaller population of humans, then in several hundred individuals. Again, systemic and local side effects are seen and understood. The immunogenicity is understood for humans, and then it will be done for several thousands and tens of thousands of individuals. The criteria that they also need to understand is what will be the dosing, what kind of adjuvants will be used, what kind of manufacturing capacity and consistency will they have, and then what kind of regulations will they need to meet. And last but not the least, how much money will be involved in all of these phases. And at the end of it, if none of this vaccines goes to the approval phase, then there will be so much capital and so much effort lost at the end of it. So this is just a picture of me and a coworker wearing the very not comfortable N95 mask, going about our business in the labs. So that's all I have, and thank you very much. I'll take any questions if you have.
Hello, am I audible?
Yeah,
Yes,
I can hear you.
Rohini.
Thank you, ma'am, for an enlightening and insightful presentation on vaccine development process. We are grateful for the time and effort you took to share your thoughts and experiences with our participants. Now we move on to question and answer session. Ma'am, there's a question from Pushkar. Is it compulsory to carry out phase 4 clinical trials for viral vaccine or only bacterial origin?
It actually really depends on the discretion of the company. That's just what I think. If there is any advancements in kind of the formulation or anything that you can do after this vaccine has been, licensed, then a company can go ahead and do a phase 4 trial, definitely. And it can be done for bacterial as well as viral platforms.
Okay, next question is from SK Das. There are many viruses. Why no vaccines are not developed since long time? Research says 50 to 60 years.
I'm sorry, so what is the question?
There are many viruses. Why no vaccines are not developed since long time? Research says 50 to 60 years.
Okay, so why there are no vaccines available? I don't know. I mean, from my experience of working, I'll give you an example of RSV vaccine, right? So, respiratory syncytial virus is a virus that is very prevalent and it affects millions of kids worldwide over a period of a year. And the investigation for that kind of a vaccine was done at Pfizer itself since the 1980s. And the success rate was just not good enough. The regulatory authorities just don't take minimal efficacy or a minimal safety candidate. So that's my understanding of why it's so hard to get a very efficacious and a definitely safe vaccine. And that's one of the reasons why some of these viruses don't have vaccines yet. Another is some of the viruses that are out there, it's really difficult to understand how to mount an immune response against it. For example, the HIV virus, it is a retrovirus and it is capable of incorporating into the DNA of a human. So how to deal with such kind of a virus? There is very little known about, the complex mechanisms that happen in such a scenario. So that is another reason why it might take forever. First to understand the virus itself and then to come up with the strategy to counter it. Are there any more questions?
Next question is from Rucha Pawar. Slide 23. What is the neutralizing factor which you mentioned in the graph? Hello? Ma'am, ma'am, please unmute yourself.
I'm sorry.
Yeah.
Okay, can you see my slides? Are you able to see my slides?
No, ma'am.
Okay, hold on. Okay, I'm having some technical difficulty sharing my screen again. Anyway, I'll explain. So you wanted to understand the neutralizing titer a little more in detail? So in the neutralization experiment, what is done is basically either the person or an animal receives the vaccine, and then the person will generate antibodies against that particular antigen that is in the vaccine. These antibodies will then be put together in a reaction with the virus itself. If these antibodies can bind and neutralize this virus, then this virus won't be able to produce any kind of infection. And that's what typically happens in a neutralization experiment. Are there any more questions?
Yeah, ma'am. Next question is by Gurpreet Singh. Is aluminium adjuvant safe to use, as aluminium nanoparticles have high affinity to bind to organs and can make cellular damages?
I'm actually not too familiar with aluminum itself as an adjuvant, but I mean, especially in nanoparticle formulation, all that I know is that there are several different approved vaccines out there that do use aluminum, but I'm not really sure about the nanoparticle formulation, to be very honest.
Next question is by SK Das. How many days we have to wait for vaccines against COVID-19?
So that was one of the annoying questions that I told you that is recently asked to scientists worldwide. I mean, there is no concrete answer. Recently, the Pfizer's CEO gave a statement that we could have some positive data for our phase 3 trial that's currently ongoing towards the end of this month. So fingers crossed, hopefully in the next few months, that's my hope.
Okay, next question is by Anita Yadav. What is the process of finding most appropriate antigen for any vaccine?
So like I showed you the structural modeling in one of my slides. So basically what we try to understand is what kind of surface proteins are responsible in the entry mechanism of a particular virus or bacteria into the host cells. So they'll see these interactions, they'll basically use this virus in a cell culture model, and then they'll see what kind of interactions happen with the electron micrograph or scanning electron micrography images to see what proteins of this virus or bacteria interact with these receptors that are on the cells, like Fc receptors or ACE2 receptors, etc. And based on that, the antigens are picked. So the basic entry mechanism of this virus into host cell is what is blocked. That is something that we use.
Next question is by Raju Gupta. Can you summarize the clinical trials happening in the process of development of vaccine?
Yeah, so I think if you remember, I just had a brief slide on it. I can go back. So these are all the approaches. Can you see my slides?
No, ma'am.
Okay. Anyway, so I don't know, there's some technical difficulty and I'm not able to share my screen, but so basically, several different kinds that I mentioned to you before, the live attenuated as well as inactivated ones are some of the approaches that are being used. There are some DNA vaccines that are being tested. There are some RNA ones that are being tested. Some protein VLP like approaches that are being tested. And finally, recombinant proteins is another approach that is being used.
Okay. Next question is by Matthew DeMello. When do you expect vaccine approval from FDA?
It really depends. So again, like I said, the CEO of Pfizer declared recently in one of his statements that the data, the immunogenicity data from the phase 3 trials that we are currently conducting will come towards the end of October. And then the FDA has requested certain additional safety testing to be done, which will take it further into November. So if all goes well, hopefully by the end of November or beginning of December. That's just my hope and prediction, but I cannot say anything concrete.
Okay, next question is by Joseph John. After the discovery of vaccine, what would be the time it takes to reach the normal people?
Those kind of decisions are, at least in the United States, are left to the government. Currently, what I know is that the government has purchased several billions of doses from different pharmaceutical companies that are leading this COVID-19 effort. So as soon as the vaccine is ready, we will just be handing these doses to the government, and then it will be the discretion of the government whether they want to give these to the more susceptible populations like doctors or army or if they want to just make it open source and give it to all. So it's really the government's decision. But I think what my prediction would be that they would give it first to the frontline workers and then to the more susceptible populations like older adults, etc., before it gets openly available for all.
Next question is by Shri Devarika. How do you screen the best candidate for vaccine production? What I mean is, why does your company or research lab choose to go for RNA vaccine if there are other options as well?
Yeah, that's a very good question, actually. So RNA vaccines actually are not out there. There is no single RNA vaccine that has still been licensed in the United States or anywhere in the world. The reason why we picked RNA vaccine platform is because first thing is the bulk. If you want to make a protein-based vaccine or a live attenuated-based vaccine, you need these huge bioreactors where you have to grow the virus and isolate the protein or inactivate this virus and use that as a vaccine. But for something like DNA or RNA, you can synthetically manufacture it just by knowing the code. You can put the code together and there you have your DNA or RNA vaccine. Also, it's very easy if there is any strain shift. For example, when the COVID pandemic began in January or in December last year, there was a particular strain of COVID that was prevalent. But if you track the changes of the gene sequence over time, now the strain has changed a little bit. So if you use a technology like RNA technology, then these changes can be made effectively. It is very hard to go back and make these changes in the protein-based or a live attenuated kind of approach. So that's why RNA technology is really beneficial.
Thank you, ma'am. Thank you for the informative session. Participants' feedback link will be posted in the chat box soon. I now hand over the session to Karishma Savardekar for a vote of thanks. Thank you.
Thank you.
Thank you, Rohini ma'am. Before moving ahead, an important note for participant that the feedback link is posted or will be posted on the chat box. Kindly fill it, Participants, you all will receive your certificate within 1 week. Moving to the end of the session, I, Ms. Karishma Savardekar, on behalf of organizing team of Department of Microbiology and Biotechnology of Patkar-Varde College, give me an immense pleasure to propose a vote of thanks to all those who made this webinar a resounding one. Firstly, I would like to propose vote of thanks to our college principal, Dr. Shrikant Sawant, for gracing the international webinar Biofacet vaccine development process. Thank you, sir, for the interesting and thoughtful speech which gave a beautiful opening to this webinar. I would extend a vote of thanks to the speaker, Ms. Larissa Falcao from Pfizer, for not only sparing her time but also for enlightening all of us on the topic of the webinar. Thank you so much, ma'am. I would like to express profound gratitude to our college Chief Education Officer, CEO, that is Dr. Mala Karkar, Chief Coordinator, Ms. Ruchita Rane, and Co-Coordinator, Ms. Anupama Tomar and Neha Kolasko for their moral support and guidance. We are also grateful to our sponsoring organization, RUSA. This event wouldn't have been successful without the constant support from our staff member and wouldn't have run smoothly without the technical team. So I thank you them. Last but not the least, thanking the wonderful participant who has turned up in such a great number from all over the country and for their active participation throughout the webinar. Thank you. Once again, thanks to all for their cooperation. Thank you and stay safe.
Thank you.
Hello, Larissa ma'am.
Hi, Ruchita ma'am.
Yes, thank you, ma'am. Thank you so much.
Yeah, you're welcome. It was
Yeah.
great.
Okay, ma'am.
Thank you. Bye.
I'll message you on WhatsApp now, ma'am.
Okay, sure.
Yeah, thank you.